My name is Lalita Ramakrishnan, and I'm a professor of immunology and infectious diseases at the University of Cambridge. I work on tuberculosis, and I'm going to introduce you to this disease and share with you some vignettes about the curious pathogenic personality of its causative agent, Mycobacterium tuberculosis, which we often also call the Tubercle bacillus. Now, tuberculosis is mostly a disease of the lungs. And what do you think of when you think of a TB patient? You think of someone who's thin, very thin, emaciated, and in fact, the disease has been called consumption over the ages. People also tend to have fevers, loss of appetite, they cough a lot, and often, as you can see here, they cough up blood. If you were to look at an X-ray of their chest, you would see that their lung is ravaged by TB as well. You can see a big cavity in there that is teeming with bacteria that they're coughing up in that sputum there. Now, while we often think of TB as a lung disease, in fact, TB can affect multiple organs. So on that upper left panel there, you see a lung that has been ravaged by TB. But you can see here that many other organs, pretty much every organ in the body can be affected by TB. The only thing is that TB, as I have taught my medical students for decades now, is transmitted through the lungs. As you can see here, the bacteria spew out of an infected patient and land in the lungs of an unfortunate individual who happens to be next to this person. So TB in any other organ is going to be a dead-end disease. And so in terms of the pathogen's survival, it doesn't do very much for the pathogen. Now, TB was discovered by these two physicians. Jean-Antoine Villemain was a French physician who first identified TB to be an infectious disease. And then Robert Koch really elaborated on this elegantly in 1882. So before this, for many centuries, people had recognized, well, not centuries, even millennia, people had recognized that TB was a single disease entity. The ancient Greeks knew this, for example. But it's these two gentlemen who identified it as an infectious agent, and then Koch actually figured out that the disease was caused by this particular bacterium. Now, at the time, in the late 1800s, when these when Villemain and Koch were making these important discoveries, TB was a terrible problem in Europe. So a seventh of Europe's population was dying of this disease, and a quarter of the working adults of Europe were dying of TB. Now imagine how that would be to have a quarter of the workforce decimated by a single infectious disease. And in fact, we all know that there are many famous people who died of TB, and I'm showing you some of their pictures here. And you can see that these are all young European men. Well, one of them, the guy on the right there, Ramanujan, is obviously not a young European man. He was an Indian man. But it turns out that he was a mathematical genius who was recognized by a mathematician in Cambridge, a guy called G.H. Hardy. And Hardy invited Ramanujan to come to Cambridge and do some math with him. And that's when Ramanujan came down with TB, and as you can see, died some years later of it. Now, we think of TB as a disease of the past, something that opera singers got, and poets and musicians of the past, and maybe something that our grandparents had, or if you're older like me, your parents. So we here in North America and Europe really don't think of TB as something that is something to worry about anymore. And in fact, people will often ask me, is TB, oh, you work on TB? Is TB back now? But actually, TB never went away. And in fact, there's more TB now than there ever was before. And as you can see, it's concentrated in certain parts of the world, in Africa, as you can see, and pretty much all of Asia, in Europe. We have Russia that's affected by TB, and then so is South America. And this is very sad. And so it's important to stop for a minute and think about why this might be the case. And in my view, there are two reasons for this. The first is socioeconomic. So TB is a disease that disproportionately affects the poor. And this is because of the fact that it transmits best in crowded conditions with poor ventilation. So for example, your urban shanty town would be a place where TB would spread a lot. In terms of who gets TB, malnutrition is a major risk factor, cigarette smoking is a risk factor, environmental smoke, so smoke from cooking in these crowded, unventilated environments is a risk factor. And then there are more modern risk factors such as diabetes and also HIV. And so about 35% of the TB in the world today is amongst people who have HIV. From a medical standpoint, there are issues that have made TB difficult to eradicate. So for one thing, we don't have an effective vaccine against TB. There's a vaccine that was developed in 1921. I'll tell you a little more about it later, but it's basically not that effective. In terms of antibiotics, we've had antibiotics since 1950. And in fact, the 4 antibiotics that we use today were all developed between 1952 and 1962. But the problem is that to reliably cure TB, you need to treat a person with 3 to 4 antibiotics for 6 months. Now, any one of you who's tried to take antibiotics even for a week will realize how hard that is. When you start to feel better, you stop taking antibiotics. Now, imagine if you had to do this in a place where you didn't have great access to health care, and perhaps you had to trek a long way to get your antibiotics, and it meant losing a day's work. So it's easy to see why people stop taking medicines when they start to feel better. And then, unfortunately, what happens then is that the disease relapses, and they get contagious disease again. And perhaps it's because of this that we now not only have TB persisting in the world, but we also have drug-resistant TB. And you can see that that has affected many parts of the world. And drug-resistant TB comes in many flavors, depending on its extent. So if the bacterium is resistant to just the frontline drugs that I told you about, rifampicin and isoniazid, then it's called multidrug resistant TB, and then you have to treat the TB with other drugs that are not as good for sometimes as long as 18 months. But then you can get what is called extensively drug-resistant TB or even totally drug-resistant TB, which is basically a death sentence. And so not only has the problem not gone away, but the problem has been compounded by an alarming rise of resistant TB. And what is even more scary is that this extensively drug-resistant TB is perfectly able to transmit from individual to individual. So it's a very good infectious agent. So let's have a quick look at the life cycle, the so-called life cycle of TB. So as I've alluded to, it's transmitted from person to person. So a person coughs it up, it lands in the lung of the unfortunate individual next to them, and then it gets into these cells that are called macrophages, and then tricks the macrophage into taking it in and forms these big aggregates that we call tubercles. And then it has to break out of the tubercle to get out again and be contagious. So here is a fundamental difference between TB and some of the world's other great bacterial killers. TB is completely dependent on producing active disease in the host in order to transmit. So it's sort of an obligate pathogen, and I use that word in a slightly different sense than other people do at times. Whereas if you think about other great bacterial killers, such as, let's take the instance of plague. Plague is really not it's an accidental human disease. So this infection that has decimated humanity over the years is really an accident. And human infection has no relevance to the evolutionary survival of the plague bacillus. This is even true for things like the pneumonia bacterium, the pneumococcus, or the meningococcus that causes devastating meningitis, or your strep-eating streptococcus. In all of these cases, these bacteria are just mucosal colonizers. They live in our mucosal tracts, and disease is occasional and accidental, devastating as it is. Not so for TB. It needs to produce disease in order to transmit and survive evolutionarily. And perhaps this explains why there's a curious feature that TB has, and that is it lacks what we call these classical virulence factors. It lacks capsules that bacteria have to avoid being eaten by a macrophage, flagella that bacteria use for motility, pili, toxins. Pili are used for adhesion. Toxins are used, they're basically they poison the host cell. And this is all talked about very nicely both by Stanley Falco and also by Ralph Isberg in prior eye biology talks. So TB doesn't have any of these. So how is it so successful? Well, there's a few things, and I'm going to tell you about a couple of them. So one thing it does have is this waxy coat that, as you can see, gives the colonies a characteristic appearance. And then if you were to stain the bacterium, that stain there is actually taken from a patient sputum, you'll see that it stains in a characteristic way with a very specific stain, and we call them acid-fast bacilli, or some people call them red snappers. And this is because of that complex cell wall. So let's have a quick look at that cell wall. So if you look at this cartoon, there's a layer just above the cytoplasmic membrane that is the peptidoglycan that pretty much all bacteria share. But then you can see that above it, it's got a really complex lipid array of lipids. Some of these lipids are complex to sugars, others are complex to proteins, and these lipids seem to contribute a lot to the ability of mycobacteria to evade the host and sort of and be pathogens. And I'm going to illustrate this for you with a discovery that was made in my lab by a PhD student, CJ Cambier. So CJ wanted to really look at that very early event of TB. So after it gets into a host, how does it get into a macrophage and survive? And macrophages are primary immune defense cells, and their job is to come to bacteria and eat them and kill them. And here's a video of a macrophage in culture eating bacteria. I've basically taken this video from Manuel Amieva at Stanford, who also gave it to Stanley Falco. So you'll see this video in Stanley Falco's eye biology lecture as well. And you can see these macrophages reaching out and eating bacteria. And Stanley refers to this as macrophages eating peanuts from a bowl. And now, when bacteria get in to macrophages, your garden variety bacterium is killed. And Stanley also talked about this in his lecture. The way this happens is that the bacteria have on their surface you see those funny little protrusions? Those are called PAMPs, for pathogen-activated molecular patterns. And these PAMPs activate in the host a pathway a signaling pathway called the toll-like receptor signaling pathway. And this pathway brings macrophages to the bacteria, and they'll eat them up, and then they can kill them using various microbicidal mechanisms. So you can see how if a mucosal pathogen is in the right place for the host, as in on the outside of the mucosa, the host might let it be. But if it tried to get in or sort of started to wander, the host would send out these macrophages that would then kill it. Now, I've already told you that TB has to get in. It has to traverse this barrier to get in. It doesn't want to hang out with these pesky commensals. It wants to get away from them and deal solely with the host. And so, but TB also has a ton of these PAMPs. So how does it do this? And what CJ found was that it uses some of these cell surface lipids to coat the PAMPs. So the blue lipids here are something called PDIM, and PDIM coats these PAMPs. So now it prevents TLR-mediated cell migration and engulfment. But then it still needs to get in. So how does it do that? Well, it adds on another lipid, and this lipid is a phenolic glycolipid. And this phenolic glycolipid brings in a new and different kind of macrophage that is not microbicidal. It can engulf the bacteria, but it's permissive to the growth of the bacteria. So in other words, mycobacteria telling the host, thank you, don't worry about coming along with your macrophages. I'll bring in a different kind of macrophage from you that can help me get in and survive. So here, just to reiterate, your bacteria on the left are your garden variety bacteria that will get killed by microbicidal macrophages that are recruited by the toll-like receptor pathway. The mycobacteria don't use that pathway. They hide from it using these lipids, and then instead they turn on, they activate the production of a chemokine that recruits these permissive macrophages that, as you can see, take up the bacteria and then take them inside. Now, this is a very this use of these two lipids is a very nice evasion strategy. But if you think about it, there's a problem with this, and the problem is that we inhale TB and into our nasopharynx, and our nasopharynx is full of bacteria that trigger TLR signaling. So even if TB has even if Mycobacterium has a way to evade toll-like signaling, there's going to be a lot of toll-like signaling going on right there. And so it would be basically caught in the crossfire in this battle between the host and the bacterium and essentially be collateral damage. So the bug has evolved a trick for this, and that is that unlike a lot of respiratory pathogens that are transmitted through the upper respiratory mucosa, TB goes deep down inside, and that's again illustrated in this cartoon you've seen before. But watch those red droplets, right? They go deep down into the alveoli of the lung. So TB is not a disease of the upper respiratory tract. It has to go deep down in small droplets that contain maybe 1 at the most 10 bacteria. And this has been known for quite some time, both through human epidemiological studies of how TB spreads and also through animal models. And this is a very nice experiment that illustrates this. So these researchers at Johns Hopkins University in 1948 gave rabbits TB, and they collaborated with some engineers. And what they did was to devise a way to give rabbits TB so that in large droplets that contain 10,000 bacteria. And these bacteria got stuck in the upper airway. They would follow them just by killing rabbits and transecting them and seeing where the bacteria landed. They also then took bacteria and put them in small droplets so that 1 to 3 bacteria were given to the rabbits, and they showed that these droplets went to the bottom of the lung, to those alveoli that I talked about. And then they followed what happened to the rabbits. And you can see that the rabbits that got more bacteria didn't get infected, whereas the rabbits that got fewer bacteria got infected. And so TB has known that more is not better, less is better, and has devised an additional strategy. So it has to use 2 lipids, and it has to minimize its droplet size. And so I illustrate for you here how TB escapes from the lower air, from the other commensals in the, from the commensal, sorry, in the upper airway, and goes down to the lower airway where it's all by itself and can do this recruiting trick using its lipids. Now, this has, there's a couple of instructive points here. One is that TB is less infectious because it has to use the small droplet strategy. It's actually not nearly as infectious as a common cold or measles or other things that have to transmit in the upper airway. Conversely, we could think about it as the commensal flora being protective against TB, as we're finding more and more that they are against many infections, and TB doesn't seem to be an exception. But finally, TB has managed with this strategy and has been around since before the Neolithic demographic transition. It's been around for something like 70,000 years. So the strategy is working very well for it. So these 2 lipids that I just finished telling you about turn out to be very important determinants in the evolution of pathogenesis. So pathogenic mycobacteria evolved from soil-dwelling mycobacteria, and the acquisition of these lipids was a major part of that step. But when we move on to now look at the next phase of the life cycle of these bacteria, which is living within this macrophage they've recruited and going on to form the granuloma, there's a little bit of a surprise, because it turns out that the determinants that are used to survive in the macrophage, bona fide virulence determinants that are studied quite a lot, turn out to be also present in the soil-dwelling bacteria. And it looks like they've just been repurposed from soil-dwelling bacteria to confer virulence. And one such example is the bacterial efflux pump, which we don't know exactly what it does in the soil, but it's very possible that this pump was used or is used in the soil-dwelling mycobacteria to pump out antimicrobials or antibiotic-like molecules that other soil-dwelling organisms put out, because it's a warfare out there between these different organisms in the soil, and that's why a lot of antibiotics were discovered in soil-dwelling organisms to protect themselves against other organisms. And this insight came from a graduate student in the lab, Kristen Adams. And what Kristen found was that these bacterial efflux pumps got induced in the pathogenic mycobacteria when they went into macrophages, transcriptionally induced. And they then protected the bacteria against the macrophage and allowed them to survive in the macrophage. So they were macrophage-induced virulence factors. So but here's the twist. When Kristen looked a little bit more at this, she found that they also mediate a phenomenon called bacterial tolerance. So antibiotic tolerance. Antibiotic tolerance is a phenomenon where the bacterium doesn't become genetically resistant to an antibiotic, but nevertheless is phenotypically resistant to the antibiotic in the absence of any genetic resistance. And it's often induced by particular environmental conditions. This phenomenon of tolerance has been known for a long time and has been thought to be the reason that TB takes so long to treat. But the model that has been proposed for this is that when bacteria enter the host and become part of the host granuloma, the tubercle, they essentially become dormant. They undergo metabolic and a replicative arrest, and as a result, they become resistant as intolerant to the antibiotics that typically tend to target bacterial determinants that are needed by actively growing bacteria, for example, your cell wall or your ribosome or your transcriptional machinery. And so in this model, the most slowly growing bacteria would be the ones that would be the most tolerant. And a lot of attempts to make new antibiotics to shorten TB treatment are predicated on this model. However, when Kristen looked, she found that these same pumps that allow the bacteria to survive in the macrophage also mediate tolerance against frontline antibiotics used for TB. And in fact, every antibiotic that she tested the bacteria underwent tolerance to that antibiotic upon entering a macrophage. So this has a profound clinical implication because now in her model or in her observations, the most rapidly growing bacteria are the ones that are most tolerant to the antibiotic, presumably because they're pumping it out. And there's a clinical in here with this finding because there are efflux pump inhibitors available, bacterial efflux pump inhibitors. Many of these just happen to be drugs that are around for other purposes. And the one that we honed in on, or Kristen honed in on, was a drug called verapamil, which is a calcium channel blocker that's used to treat high blood pressure and cardiac arrhythmias and migraines. And she found that if she treated the macrophages that were infected with verapamil along with the standard chemotherapy, they were killed much better with standard chemotherapy. And based on these results, a clinical trial is going to start in India to see if verapamil will shorten the course of treatment. From an evolutionary standpoint, it's kind of interesting because we're talking about a pump that was used and is used in soil-dwelling mycobacteria, presumably to pump out antibiotics. Then over the course of most of the history of pathogenic mycobacteria, it was repurposed to fight macrophage defenses. And then in the last 100 years or so, since the advent of, or less actually, 50 years or so since the advent of chemotherapy, that ancestral function has come back to help the bacterium withstand the modern post-antibiotic era. Moving along, we've got the bacteria now living in what is called a granuloma or a tubercle. And while the initial cells that come and form this granuloma are macrophages, the body then brings in, the immune system brings in many more different cells to come in and help fight the bacteria, and yet in a proportion of cases, the bacterium is still able to survive in this rather complex structure. And this brings me to a point that I want to make, which is that you'll hear many times people say, and you'll read in books, that a third of the world is infected with TB. And the idea there is that they're infected with TB, they've got it under control, and then but at any given time they can reactivate this latent disease and get morbid and transmissible TB. But actually, this is based on the TB skin test, which simply tests to see if you've ever been infected with TB. And so the fact that your skin tests positive doesn't mean you have TB infection now. It means you once had TB infection. You could still have it, or you could have cleared it. And if you really go and look at old epidemiological studies, old and new, you'll find that most people who get infected with TB actually clear it using this arsenal of immune defenses and a combination thereof that I've listed for you here. And so actually most people eradicate TB and a few people go on to have primary disease, and latency and reactivation disease are no doubt present, but in my view, based on my reading of the epidemiological studies, are a minority of cases. And this is very nicely shown here in a study done in Amsterdam, where contacts of active TB sufferers were followed meticulously, and if they got TB at all, they got it within the first few months of exposure. So the immune system does a pretty good job of killing TB. And if that's the case, then why don't we have a vaccine for TB? A vaccine for TB has been the holy grail. But in fact, a vaccine for TB was made and was first administered in 1921. It was a live attenuated vaccine. And we still use it to this day in highly endemic areas because it does offer a modicum of protection against disseminated TB and meningeal TB in kids, which, as you'll see in my third lecture, is a terrible disease. But obviously, we still have a lot of TB, though it's the world's most widely administered vaccine, and that means it doesn't protect very well at all. And efforts to improve its efficacy or to make whole new vaccines have not worked very well. So why is that? Well, that's a paradox. We don't really understand why, but I'd like to tell you a few things. First of all, most people don't get TB when they're infected. They're protected naturally. But if you do get TB, then you're not completely protected against a second infection, which is different from the case of, say, smallpox, where if you get smallpox once, then you don't get it again. People can get TB again. On the other hand, there's very clear data that show that if your skin tests positive but never manifested disease, you're somewhat protected. And that's nicely shown here in a study of nurses in Norway who in the pre-antibiotic era were found to be either skin test positive and skin test negative, and then they were put amongst the TB patients to take care of them. And the ones who were skin test negative were much more likely to get TB. So the people who were skin test positive but had never manifested TB were somewhat protected against TB. And if we can understand this, look at these data again with new eyes and understand them a bit better, maybe we can try to think about ways to recapitulate the protection that many people in fact seem to have. So in closing, we've got a bacterium here that is possibly one of the world's most successful bacteria that evolved from non-pathogenic environmental mycobacteria, doesn't really have a great many classical virulence factors, but seems to use a sort of stealth mechanism to survive. And even though it doesn't produce disease in most of the people that it infects, that's good enough for it because it produces just enough disease to have survived over the eons. That is not to minimize the impact of TB. TB killed nearly 2 million people last year or in 2015 and caused disease that debilitated them in about 10 million people. And perhaps the most chilling thing to consider is that TB's been with us for more than 70,000 years, and it's still with us. It predates the transition to an agrarian culture. It resisted urbanization. In fact, it thrived with urbanization. It's resisted modern medical technologies like antibiotics and attempts to make a vaccine. And so it's a pretty tough and wily bacterium. And if you stay tuned for parts 2 and 3, I'll tell you about some more insights we've had about this bacterium using a very interesting and unusual model to derive these insights. Okay, thank you.